for the introduction, Angel, and also thank you for giving us this opportunity and to present our teamwork for the study of the COVID-19. And in this study, we are focusing on the structural changes of the SARS-CoV-2 spike protein at different temperature, pH values, and also saline concentrations. We are using both the computational methods as well as the experimental methods, and that can help us to validate our numerical predictions. So in our today's talk, we will just present our methods as well as some key results. And if the audience have quite interested in our research methods or research data, we can talk separately. So first of all, why do we need the spike protein? The SARS-CoV-2 spike protein is the key receptor for binding to different material surfaces, and also for promoting and the entry to the host cells. So, and this fact is quite correlated with the efficiency of the COVID-19 spread between human and human, even without contact. So far, we know the structure of the spike protein, and also we know the binding site of the protein to the host cells. So what we need to learn from this study is the structural changes of the spike protein as a function of different temperatures, pH values, and salt concentrations. So for example, if we know how long the virus can survive on top of the surface, and how infectious and different healthcare conditions such as temperature, pH value, and such concentrations, we can better protect ourselves from the infections, and also we can find out better ways and to clean the virus from our environments to protect us. So in today's talk, and we would like to present our simulation results and also in vitro experiments results that can help us to validate our numerical predictions over there. So first of all, and I use this flowchart and briefly show our research pipeline. We start with the molecular dynamics using the atoms to present the structures of the proteins in the water system. During the simulations, we collect the trajectory data, including the positions and velocities. Before analysis, and also we performed the structural alignment method. Next, we conducted the data analysis of the simulation and the trajectory in terms of the MSD, root mean square deviation of the atomic positions, and the SOSA, solvent accessible surface area, as well as the hydrogen bonds between the proteins and the water molecules. Next, we also focus on the RBD analysis, receptor binding domain analysis. And we want to study how the RBD areas structure changes and at different conditions or combined all those conditions. Last, and also we conducted and further analysis and based on the residues and the chains, so that we can just show which groups of the residues contributed the most to the structural deviations and during the simulation. So first of all, and I want to show the MSD results and for the spike protein at different temperatures. So here and in the top right and the figures, and I showed the MSD values and for the temperature between 3 degrees and also as high as the 95 degrees. So, and our simulations last 3 microseconds. So here we already delete some equilibrium warm up phases. So we can see that as the temperature increases and the MSD values and also monotonically increased over there. And in the lower left figures, and we show 3 extremely high temperatures, which are the 150, 200, and 300. We want to use those high temperatures and to test the limit of the molecular dynamics, as well as that's learn and speed up the denaturing and the phenomena of the virus and as extremely high temperatures. Also, we can see the movies and the structures is already totally damaged at the high temperatures. So let's focus on the MSD values and for the temperature between 3 and 95 degrees. And we can, by our naked eyes, we can see three groups over there. One, the bottom one is the low temperatures, and for the 3 degree, and the middle one is kind of the normal temperatures between 20 and 60. For some high, for some high temperatures and beyond 70, and there are the last group of the temperatures over there. And also to be intuitive, and also we superimposed the molecular structures of the different temperatures. And also here, the control group is the normal temperature 37 degree. And here we use the colors and to present, to show the structural deviations in terms of the nanometers. So we can see that as we increase the temperature, not only the MSD and is increasing, and also we have more and more means that shows larger deviations and from a few angstrom to nanometers. So here and in this figure, we just compared all of the available MSD values for different temperatures. We can easily see and the monotonic increasing trend and from the angstrom and to the nanometers and the range. In addition to the MSD values, and also we calculated the hydrogen bonds between the protein and the water molecules. So in this study, and in this measurement, we also can see that as the temperature increases over there, and the number of the protein water hydrogen bonds is also monotonically decreasing. And also here and in the lower left, and I show a figure and for the distributions of for the protein water hydrogen bonds and using 200 nanoseconds of the data. And also we can see that the higher temperature, the least number of the hydrogen bonds and between the protein and the water molecules. And in addition to the protein water hydrogen bonds, and also we studied the hydrogen bonds between the mean chain and the mean chain. And generally speaking, as the temperature increases, and the hydrogen bonds for both components and decreasing. However, and the decreased speed for the protein water is much larger, bigger, and than that for the mean chain to mean chain hydrogen bonds over there. So this is another measurement. So I will we showed for the SALSA. SALSA is the solvent accessible surface air, and also we can see the the same trends over there as we increase the the temperatures and the SALSA area and monotonically and decreasing and dramatically. And this is the MSD and the distributions, and show the decrease of the MSD values as with the increase of the temperatures. So first of all, and we calculated the correlation matrix and between the different measurements, including the MSD, SALSA, and the hydrogen bonds. We can see that the very strong correlation and between those values. And the last, and we are using the k-means clustering and methods, and using the data we collected from the molecular dynamics simulations, and to classify the different temperatures. Interestingly, and we found out 4 groups of the temperatures. For example, let's look at the first group. The first group is kind of the 3 degree. 3 degree and is kind of the temperature in the cold supply chain and the systems. And also, and in the literature and from the clinical part, and we see that at the low temperature, such as 3 or 4 degrees, and the virus can survive for and up to 2 weeks. And for some normal temperatures, we classified over there, and we can see that, and in that range, and the virus could survive for a few days. And in the a little high temperature, 60, and we found out that this is kind of the critical temperature and the pure rate. And even at that temperature, and the virus can also survive like for minutes. So that is still not very safe. For some extremely high temperature and above 70, and the virus could be killed in just a few seconds. And using our large amounts of the molecular data, and not only we can get the whole protein analysis, and also we can just focus on some chain-based or residue-based analysis. And further, and we can just show the microstructure changes of the spike protein at different temperatures. And later, I will just show some experimental results and to validate our numerical discoveries here. So first of all, and I want to show the H1N1 and the virus temperature test, which is conducted by the Dr. Feng and the Professor and Marian. So here, and because the COVID-19 virus is still at the high risk, so that we use the H1N1 virus instead. And after a half hour of the constant water bath, and we can see the log reduction of the virus in the environment. As we know, the 6 log kill rate means if the log reduction number is more than 6, 99% of the virus will be killed. So we can see that the critical temperature and for the H1N1 virus is between kind of between the 51 and 53. And also here we added more data points in the experiments. We found we proved the monotonic and increase and in the log reduction as we increase the temperatures. And more interestingly, and also we just change the exposure time. We found out that the exposure time to the high temperature is also quite influenced the number for the log reduction. For example, if we reduce the exposure time from half hour to 10 minutes, means we need a higher temperature and to kill the virus. So actually here we gather that if we plan to kill the SARS-CoV-19 virus and in less than a minute or in a few seconds, we may consider the high temperature as we found out for the beyond the 60 over there. And considering the time, and in the following and slides, I just briefly and talk about present our key results over there. So here, and besides the temperature tests, and also we conducted the experiments for different salt concentrations. For example, this is the MSD salsa, and as well as hydrogen bonds for the spike protein at different salt concentrations. In this slide, we studied 3 different salt concentrations, and between 0.5 molar and 1.5 molars. Actually, and the sum of the conclusion is that these computer simulations, as the increasing and the salt concentrations, didn't suggest very big and structural changes, and that could be influenced by the salt concentrations. So actually, and on our experimental team, and doing the same experiments for different saline for the H1N1 virus at different saline waters. And we still remember the 6 log kill rate. We can see that as we increase the salt concentration and from 0 to 5, and the H1N1 virus is not sufficiently and killed. Also, it means that okay, and we guess that it may be not safe and go to the beach because the highest concentration salt water actually didn't protect you and from the virus infection. So different from the salt concentrations, we found out the spike protein is more sensitive to the pH values. What is the conclusion? So here, this is the key results. And in the very high or low and pH environment, such as 3 and 11, and the structure of the virus actually changed dramatically, almost be killed over there. And so here, and this is the results, and for the MSD values and for the spike protein at pH at 3 and 11. And similarly, and on our lab bench team and conducted the experiments and for the H1N1 at different pH values. And for the low and high and pH values, and we see and a dramatic and a big value, and for the log reduction means that like a majority of the virus or already killed in that case. So here I want to give you an analogy and to those like pH values. For the pH value between kind of 4 or 5 is kind of the values for the apple and the beef. Actually, the virus cannot be like effectively killed at that pH values. However, for some grapes or even for some like most of the cleansers and bleach we are using, it's like can dramatically change the structures of the spike protein. That means like causing the disinfection of the SARS-CoV-2. Hope that you can just like clean under the surface of the substrate and using those kind of the pH value and things out there. Okay, let's come back to the temperature test. And in the previous slides, I showed that during according to our MD simulations, and we found out like as we increase the temperatures, actually we found out the permanent microstructure changes at different temperatures, especially for the 60. And for some high temperatures and the substructure and the changes is more dominant. But this is kind of the computer results or predictions. How can we prove that? Actually, on our lab team and doing the the imprinting and the process. So this is the principle of the molecular imprinting. The protein or virus is removed and leaving the imprint and behind. When the chip over there is exposed and to the solution containing the imprint and proteins or viruses, and actually we can collect the signals and for the existence of the proteins or virus with such with the same structures. And so here, and we did two experiments. The first experiments, we used the imprint from the normal and the spike protein. And as we increase of the protein and concentrations in the solution, and we can see that the biosensor can find out, can detect the existence of the S protein. However, for the heated S protein, and we cannot find it. And the same thing, and we just redo the experiment. So this time, and we imprint and with the 60-degree heated like a spike protein. And then we just try the solutions, and for the two kinds of protein, one is the normal, the other one is the heated like a spike protein. And as we increase the concentration of the virus, we can see that the heated spike protein is quite detected. So this is the kind of the technique we can use to prove the permanent structure and difference occurred at the high temperature 60 and in the spike protein. In other words, and this kind of experimental results confirmed and our model and the predictions and engraved the experiments and the reporting and the inactivation of the H1N1 and the SARS-CoV-19 COVID-19 viruses over there. So last, I want to use 2 minutes and talk about our larger computer simulation systems. As Joe mentioned and previously, and we are studying the blood class. And also we found out the blood class are quite common in the patients and with COVID-19. For example, we already found out 20% of the COVID-19 patients can be found the blood class. And this number increased to the 31% and for the ICU and the patients over there. So right now we have conducted the simulations for the 125 million particles and to simulate the platelets. Here we have 250 and platelets and flowing in the blood vessels. And using the molecular dynamics, we can just extract the physics modeling based coefficients and study the binding and between the cell molecules and to the blood vessels. And our goal is that we want to find out and how fast and how strong and the thrombus can be formed and attached to the blood vessels. That may like eventually block or the like transform of the blood and through the blood vessels. So previous slide shows the multi-scale modeling and systems. And this slide shows that the way we do the analysis. This is a kind of the multi-scale learning and the framework. And why we want to introduce this multi-scale learning framework, the reason is that multi-scale modeling and will provide us the multi-fidelity data at different spatial and temporal scales. And we are using the different scale and learning systems and to extract the features more efficiently and effectively. So eventually, and our learning system just do the prediction and for the stability of the thrombosis microstructures. So this is our publications and for this study. So last but not least, and thank you so much again for inviting us and to present our work. So thank you.